Okay, it's 11, so we can get started. All right. Great. So I hope everyone got the readings. Sorry again for last time. This time I hope you all had enough time. Okay, so this is the third class of the module. We have one more after this. Today we're going to talk about competitive dynamics. We're going to continue talking about two species. In the last class, we were talking about this particular kind of interactions between two species, which are these anti-symmetric predator-prey interactions. And today we're going to talk about a different kind of interaction. And in fact, this is what we're going to continue talking about in the next class as well, which are perhaps even more common than these predator-prey interactions that we were speaking about in last class. And this is when you generically put two species together with each other, many a time they tend to compete with each other. And so typically when we do experiments of a large number of pairs of species, we see these three common outcomes as a result of competition. And these are really qualitative outcomes that we're talking about. It's either that one of the two species excludes the other and wins this competition. And so after a long time, even if you mix two species, you'll only see one. Or, a very interesting outcome is that both of these species can coexist with each other in some stable equilibrium state. And that, of course, allows us to talk about diversity for the first time. And that is, and we'll spend a lot of time talking about that in this class, but even more talking about it in the last class. And then finally, what can happen is you can observe this bias stability where, again, eventually the outcome is either one or the other species wins, but which species wins depends on how you initially set things up. Whereas in this exclusion outcome, no matter how you set up the initial conditions, it's only one of the two species that's going to win. And then we're going to talk about the simplest possible model of this competition, which again is, also due to Lotka and Volterra, but this is not to be confused with the predator-prey model. This is the Lotka-Volterra competition model, and here we see that the set of outcomes qualitatively that you get as a result of two species competing matches exactly these three outcomes that we see in experiments, which is nice. But there are generalized versions of this model once you allow for more than two species, which allow more outcomes. For instance, they allow oscillatory things, chaotic things, which are not necessarily stable states of this type. And that we won't talk about so much in today's class, but they'll become more important in our last class of this module. Yeah. Okay. So so this idea of competition is, of course, very, very common. In fact, Darwin, when he was thinking about the origin of species, one of the key things that he talks about in one of the chapters that he has in this book is about struggle for survival, right? And so there is a sense among most naturalists when you go out and observe the world that species actually, they're coexisting with each other. It seems kind of harmonious, but if you start to really look, there's a lot of competition, right? And it's this competition that we would like to understand better. Right? And so there are these classic experiments that go back to Gauss in the 1930s, where he really, found it easier perhaps than to mix like lions of various types or grasses of various types to mix microbes of various types and ask what happens when you mix two different species of microbes. Any idea that people have why you might want to do these experiments with microbes instead of maybe, I don't know, plants or something like that? Yeah, time scales are shorter, they grow fast, they die fast, so you could do the same things with plants, and in fact there are big ecological experiments in grasslands around the world that are being done. But if you actually go and look at those experiments, they've been going on for decades, and the time scale of learning about some competitive interaction between plants will typically be the order of years. Does someone have a sense of what the typical time scales of microbial growth are? Are they years, days, months? Hours, right? Yeah, so typically microbes, The famous number that people like to give is that we know of this bacteria that we all like called E. coli, which can, in the right conditions, grow and double once every 20 or 30 minutes. So, not every bacterium does that, but yeah, sure, doubling in about an hour or a few hours is not uncommon. Right? And so these are experiments where you can take two species of bacteria and put them together and see what happens pretty quickly in the matter of a day or a few days. And so in Gauss's experiment, what time scale did he observe these competitive dynamics over? Do people remember? Sorry? 0.5 per hour is a unit of growth rate, right? Or a death. I mean, it's a unit of rate, right? But I'm asking about a unit of time, right? How long did his experiments go on for? Right. So let's say we're trying to do this experiment ourselves, right? Let's talk a little bit about what experiment we try to do. Can someone tell me what kind of experiment Gauss did, or the other experiments that you saw in the reading? What are you doing? How are you mixing these two species and watching their dynamics? So you first need to have two species, right? Okay, where do you get them? You can extract them from somewhere. You can go to the soil, extract species. You can go to a microbiologist friend, ask them for some microbe, right? And they'll give you two different bugs. And how do you compete them? You leave them in a room and then you go away. Right, you grow them in the same medium, right? So you first grow them, you grow them to some level, and then basically you take a flask or a test tube, and in this you put a little bit of species 1 and species 2, and this is a medium which contains what? Can it contain just water? Right, so it should contain some nutrients, right? So this is a medium with some nutrients, and that's what these species both need to grow, right? And then you kind of leave them, and then what do? What happens if you leave both of these species in this medium with nutrients? Yeah, these species will grow because they'll eat up the nutrients, right? And they're probably often going to be competing for a similar set of nutrients that you provide in the medium, right? And often you only provide one kind of carbon source when you're doing these experiments, and we all need carbon to grow. So you know for sure that they're competing for this carbon, and then they'll grow. They'll grow. The nutrients will shrink, and at some point the nutrients will probably run out, and the microbes will stop growing, right? At that point, do you stop? Put another batch. What do you mean by put another batch? Right. So let's plot this. Let's say we plot on the x-axis time in our experiment, and on the y-axis we are plotting, let's say, the fraction of one of our species, right? Say a fraction of species 1, right? How do I track, by the way, how much of species 1 I have versus species 2 I have? These are microorganisms, right? How would I do that? Radioactive something. Okay, like what? Okay, do I need to do something radioactive? I mean, that then I probably can't do this experiment in a room, right? I'd probably have to go to some special chamber, wear some special clothes. Fluorescence tagging, right? What is fluorescence tagging? What's the idea here? Yeah. Right. So where should this fluorophore sit? Right. Protein in what? In both the species? Right. Yeah, exactly. So what you can do is you can fluorescently tag, basically you can color in some way each of your species. You can color species 1 in green, you can color species 2 in red or yellow, what have you, and then you can just measure the fluorescence in both of the channels in your test tube. And just by knowing the relative levels of fluorescence, you can get a sense of which species is growing more or less. Right?
But as one of them divides, why will the second one have this fluorescent one?
Why would the second one? Yeah, because you want to get a sense of how much both of them are there, right?
Once it divides, will the daughter also get the
Yeah, because the way that you're engineering this is that you're putting that some special gene inside the DNA of this bacterial species, and when it divides, it'll both daughter cells get it, and so both of them will express this colorful protein. Right? Okay, and so you're tracking the fraction of species 1, right? So what numbers can this fraction take? It can either be 0, right, which means that the species 1 is completely absent from this medium, or it can be 1, or which means the species 1 is completely taken over this medium, and then it can be something in between, right? So you start off in the beginning. Let's say you mix your species 1 and species 2 in some equal fraction. So at time t equals 0, they're probably going to be somewhere here, right? And then you do this experiment, you mix them, you let them grow. Maybe one of them will grow a little bit faster, one of them will grow a little bit slower. They'll grow and they'll deplete these nutrients. And after a while of time, a point of time, once they deplete the nutrients, will this fraction, what will happen to it? Yeah, it might have increased or decreased, right? Is it necessary that it has increased or decreased all the way to 1 or all the way to 0? No, right? So it probably increased a little bit, right? And that's over maybe one day, right? So I'm tracking time in days. And now, does this help me know what exactly is the competitive outcome of this two species competition? No, right? So what do I need to do? I need to repeat, but do I need to repeat this same thing again? Yeah, someone? You need to add nutrients, correct? Should I add them back into the same flask? This one's probably crowded with a lot of bacteria already, right? So what you do is you take a fresh flask with nutrients, test tube, right? And in this you take a little bit of this culture and then you put it in, right? And so you know that the little bit that you put it in is still going to have the same fraction that you started with, more or less, right? Because these population sizes will be gigantic. They'll be in the millions to the billions, right? So taking a little bit of that, you probably won't have that much noise. And then these can grow, right? So if species 1, for instance, continue to grow faster than species 2, this number will go up, will go up, will go up, right? So this experiment is pretty simple. I just need to every day come back, create a fresh flask of these nutrients, take a little bit of my previous culture, how much that little bit is, I can choose, right? And then I can just transfer them over and leave them be, right?
There will be equal proportion.
Should what be in equal what thing are you talking about?
Both the species should there be in equal proportion as in the initial.
Yeah, why don't you answer that? Should they be in equal proportion?
Because when we are mapping it, if you want to extrapolate the previous mapping, we need to have the same growth rate.
So I don't follow what you mean by extrapolate to the previous mapping.
Initially, we took say some x and x number of species for the species 2. Then in the new flag, should it be the same proportion?
Yeah, should it be the same? Like if you made it so that you got the same proportion from both of them, would you start here at day 1, or would you start here at day 1?
Somewhere we do.
Yeah, And then if you started here, then what would you see? You'd see the same thing again, right? And then you drop it again, you see the same thing again, right? So that of course would not help you know what's going to happen, right?
So just somewhat amount we choose when we
Yeah, you actually you want to preserve the fraction that's in this culture, right? You want to keep the same fraction that it is. So if this is 60% of species 1, 40% of species 2, you want the next class to also start with 60-40, right? So that you can ask, okay, what happens to the 60-40? Does it become 70-30? Then does it become 80-20, 90-10, 100-0? Because eventually you want to stop and ask, okay, after some point, one of these species will be lost, right? And that's might be lost, right? And that's what it might have meant for species 1 to win the competition, right? So you want to take some amount from this flask, but whatever amount you choose, if it's all well mixed, the fraction of what your fraction of each species will not change, right?
The amount of nutrients we supply would be the same.
Yeah, correct. Yeah, so you prepare the same test tube every time, basically. Yeah, so that the amount that they have access to is the same. Okay, so let's say you do this. Okay, what happened here? This this is day 2, 3, 4, 5. Maybe by the 6th day, you go in and you see there are no more species 2. Okay, well what happened? Yeah, you'd say species 1 won the competition, right? And this is what we would mean by saying species 1 excluded species 2. Okay, so that's one of the common outcomes. Okay, now, remember we started off this culture with an equal fraction of species 1 and species 2. And we saw that, okay, if we do that, then species 1 wins. Does this mean that species 1, we proved that species 1 will always win? No, right? What else could happen?
could be dependent on the initial.
It could depend on the initial condition. So can you give me a counterexample where you won't get exclusion, but you'd still see this outcome? Yeah. Okay. Let's just take the example of what happens if we instead started off our whole experiment with 60% of species 1 and 40% of species 2. We can start this off. We can do the same experiment. We can track these species. And let's say here I did this and I saw that, okay, eventually if I wait a few days, I'll see species 1 win. I did the same with 70-30 and I saw the same thing. I did the same with 80-20 and I saw the same thing. 90-10 and I saw the same thing. If I start with 100-0, then of course there's no other thing that I could see, right? Okay, now let's say I started with 40-60, right? Exclusion would mean I still see the same outcome. If I start with 30-70, 20-80, 10-90, I always, if I wait long enough, I'll always see species 1 win. But that need not have been the only thing that could have happened. So one other thing that you see, maybe for some other pair of species. Yeah?
Last time that you draw, that cannot go up, right? The fraction.
Oh yeah, of course. If you start with 0, then of course that will stay forever, right? But that's not really, that's not an outcome of competition. There was no competition that occurred there. Sorry?
Close to 0 can we go?
Yeah, so you ideally you should be able to go arbitrarily close to 0, right? Of course, at some point you'll go so close to 0 that you'll have one individual of species 1, right? And of course, going any lower would not mean anything, right? So yes, in principle, when people do these experiments, they've been able to go, 1% of species 1, 99% of species 2, and still see species 1 eventually take over, right? Okay, so just want to point out that this is not the only thing that needed to happen. So let me just construct another example. So on the y-axis, I'm still going to plot fraction of species 1, but this is, let's say some other species 1 and some other species 2, right? Here again, you might find that, okay, if you start them off at 50-50, species 1 wins. You start them off at 60-40, species 1 wins. 70-30, it wins. 80-20, it wins, and so on and so forth. If you start them off at 40-60, species 1 wins. But if you start them off at 30-70, species 2 wins. 20-80, species 2 wins. 10-90, species 2 wins. This can happen, right? So does this mean that there's always the case that one species excludes the other? Yeah, there is always one species that excludes the other, but it's not the same species every time, right? And in principle, there are two different stable states that you arrive at. Why are they stable? Right, okay, this is important. There's two aspects here, right? One is species abundances don't change after a long time. Is that enough for stability? No, right? That's just enough for a fixed point, right? That's just a steady state. But a steady state need not be stable, right? So, okay, so what would stability imply?
If you give a little bit of more population, you should come back.
A little bit of more population of what? Right? Because now there are two options. Right? You can provide a little bit of species 1, a little bit of species 2, a little bit of both of them also. So what should I provide a little bit of? Yeah, any and all should give me the same thing, right? So, I mean, the most interesting one I would say is, I mean, of course, if you had a state where species 1, adding more of it will, of course, not change the fraction. The interesting case would be if you added a little bit of species 2 to this and saw that species 2 died, right? Similarly, in this state, if you added a little bit of species 1 and you saw that it died, then you know that these are two stable states that we will obtain, right? These are our two stable states. And this is also an experiment you can do, right? Once you get each of these states, you can try invading them with a little bit of the other species and see whether or not that grows on, right? And effectively, truly this kind of experiment where you add 99% of one or 99.99% of one and 0.001% of the other is kind of like that, right? And so this kind of outcome is called bistability. This is also a commonly experimentally observed outcome. Are these the only two? There's also coexistence. And what does coexistence look like? So now I want to start off again at 50-50. And what should I end up at? If both species are to coexist, well, I better not end up at one of these points, right? I should end up somewhere in the middle, right? Do I have to end up exactly in the middle for there to be coexistence? No, right? I can end up anywhere. I could even end up at 1090 and that would also be coexistence, right? Coexistence just means if you wait a very long time, both species are present at some stable fixed point, right? And so in this case, maybe we'll see the following. Right? And of course, if you start off at that fixed point, then you'll see.
Can it be that if you start with a different ratio, you end up at it?
Great question. Right, so here in this example that I've drawn, does the initial ratio matter? Yes or no? Ready? 3, 2, 1. For the final outcome. I mean, and I don't want to take the trivial condition where you start with 100, 0, or 0, 100. Can you put up your fingers again? I just wanted to get a sense of where the... Okay, I would say most people agree that the initial ratio here doesn't matter, right? But it could have, right? So there's a fourth outcome here that I didn't draw. I'll draw now, just to point out that you don't really see it that often, though it has been observed a couple of times. So one other outcome could have been... Yeah, okay, great. You could have also seen oscillations. We never see that. To the best of my knowledge, we've never seen oscillations. We'll talk a little bit about maybe why in a second. Okay, so what you could have seen, and nothing should prevent this from happening mathematically, is the following. In some set of initial conditions, you go to a coexistence fixed point, and in some other range, you have, one of the species winning. Right? What word would you give it? Sorry? Exactly. These are also two stable states, right? And this would also be bistable. It's just that it's a different kind of bistability than this, right? So here, both the stable states, it's only one of the other species winning. Here, in one of the stable states, only species 2 is present. In one of them, both of them are coexisting, right? And in principle, nothing should stop you from seeing this, right? So this would also be a bistability. And I think we have one example experimentally where people have seen something like this. But again, this is not one of the common outcomes that we see, right? And if you were still to give words to them, these are the 3 words that you would give, right? So qualitatively, these are the 3 things that can broadly happen, right? In principle, they could have been oscillations as well. We don't see those. In principle, there could have been some weird chaotic dynamics as well. We don't see those. Yeah, this the thing to point out is this is all for 2 species. Very different things can happen for many species, which we'll talk about in the next class. Okay, so yes.
Why can't there be more than one fixed, 3 fixed points, let's say?
Oh, great. Yes. In principle, you're saying you could have had another scenario where there could have been, something where species 1, some intermediate region where there was coexistence, some other region where species 2, 1.
Or like more than one case where both coexist.
More than one? Okay, good. Yeah. So in principle, like, again, you can try and construct all of these things. So it's true. You could have seen maybe two coexistence fixed points and so on and so forth. And this is actually a great point, because now we see that there are some outcomes that we observe experimentally, but in principle, the ones that we can cook up in our head are more numerous than that, right? And so this is one of the motivations that we can have for making a simple model, and in it trying to ask, okay, what are the outcomes that we see there? And then perhaps you can understand better why some outcomes are easier to achieve than others, right? So maybe what I'm trying to say is let's put a pin in that for now. And then when we'll do the model, we'll come back and see what would it take to have outcomes like the one that you're describing. Yeah? Okay. So our task is clear now, right? We need a model where you can kind of simulate a kind of experiment like this and ask, okay, what conditions would you need for each of these outcomes to occur? Or maybe possibly other outcomes that we might see. Yes?
So in those first 2 outcomes,
Yes.
you just see species 1 wins.
This one? Yeah.
And that one.
Correct. So here species 1 wins, in these conditions species 1 wins.
You change the ratio of 2 species and species 1 still wins unless you go below certain value.
Yes, correct.
So that is, there is no way to know if it's coexistence or if it's exclusion. Sorry, if it's bistability or if it's exclusion.
In which case? So in this case, you know, if you start here, one species wins. If you start here, the other species wins. This is by stability, right?
No, I was saying you're just seeing one species wins.
Yeah.
From that you can't say if it's
Exactly.
by stability.
Yeah, precisely. So that's exactly the point that I was trying to make here. Good. That just by doing one experiment, just by looking at one trajectory, and you see one species winning, you can't say whether that means that there's exclusion or biostability. You have to look at dependence on initial conditions. That's why we draw lots of these curves.
We're also assuming determinism, right? It would be stochastic.
Yeah, correct. Yeah, in fact, if you actually look at the experimental curves, you'll see some stochasticity, right? But I mean, largely we are working in population sizes that are large enough that we're going to ignore stochastic effects. And again, if you do a whole course on this stuff, then of course later on we would talk about stochasticity.
I think the point
Yes.
was that
Yeah.
when the let's say species 1 died,
Yeah.
what if species 2 also died? We cannot make a conclusion looking only at species 1. Something of that sort. Like looking at species 1,
Yeah.
you can say, okay, beyond certain point, species 1 survived.
Yes.
Below certain point, it died.
Yes.
But without seeing species 2, how can you say that when this died, the other one survived?
Good. Yeah. Yeah, that's absolutely correct. So that's why we're taking the fraction of species 1, right? The fraction of species 1 requires us to measure the numbers of both of them, right? This is n1 over n1 plus n2. So I need to know both of them, right? Yeah, if I were just tracking the abundance of n1, I would be lost. But that I would be lost in all of these things. Okay, good. So let's talk about the model that we want to make. Okay, and the simplest model of species competition is this Lotka-Volterra competition model, not to be confused with the Lotka-Volterra prey-to-prey model. And what you're really saying here is that, let's say we do it for two species. You have n1 dot and n2 dot. And you're writing down, well, something like the following. Okay? And I'm saying that this perhaps is the simplest model that you could have written down of this kind. It's nice to spend a few minutes just talking about whenever you encounter a model like this, how do you make sense of it, right? So, okay, so first let's take the case when R1 and R2 are both positive. Why am I saying that? What if they were both negative? Then would
There cannot be growth.
Population will not grow. I mean, as the number increases, the population will decrease.
Yeah, okay. I mean, let's say for now that the population won't grow. We'll come back to this in a few minutes and see that maybe it can. But yeah, I mean, there's some sense in which it seems like the population might not grow. And so let's talk about that case. Let's say R1 and R2 are both positive. And you have a case where species 2 is not present. Okay? Species 2 not present. What the question is, will species 1 grow? Okay, let's take a vote. All right. Ready? 3, 2, 1. It's okay, we'll talk about it. I just want to take a vote first. Okay, yeah, okay. So, yeah, you're correct that this species will grow, right? and it'll grow logistically. How do we see that? You just take away n 0 and you get back logistic growth, which we have been studying before, right? Same thing with the other cases. Species 1 is not present, species 2 grows logistically, right? And of course, for that, you need R1 and R2 to be positive, okay? And maybe for the rest of the class, when we take votes, just keep the votes to yourself and we'll ask you, I'll ask you things, you'll get an opportunity to answer, right? But the whole point of these votes is for all of us to get to think about the answer before we all discuss them, right? Okay, great. So species 1 will grow, right? So that's at least one way to make sense of this equation. In the limit of the other species not being present, you just do logistic growth, right? And so in this case, where of course species 2 goes away, our sense might be that this species is at its carrying capacity. Yeah, that's fine. Okay, and now you can ask, what is the effect of species 2 being present on species 1, right? And what is that? Okay, we'll talk about the signs of beta, but just what do we say? Like, what's the model saying that there's some interaction? Correct. What form does the interaction take? Is it inhibition? Okay, let's talk about this. So, what if you want competition in this model? Wait a second. Yeah. There's no resources in this model, right? Are there? Are there any nutrients in this model?
The carrying capacity invariably kind of depicts the area.
Maybe. I don't know how that works, but this model doesn't care about that, right? Maybe it invariably represents it, and if you are interested in that, we can chat about it later after class. It turns out it's much more subtle than that. But yeah, I mean, it's clear that the variables in this model are just the abundances of these two species, right? And there are no nutrients in this model, right? So whatever we're thinking about nutrients in our head is implicit, right? Okay, so my question is the following, and we'll vote on this before we all talk with each other. What do I need to do to the betas? Do the betas need to be positive, negative, or anything? Okay, ready? 3, 2, 1. Do people not feel like voting? Okay, I would say there seems to be a mixture of options in the class. So just turn to a neighbor and talk to them about what you think is going on here. This side of the class doesn't want to talk to their neighbors. Okay, let's vote. Okay. All right, let's vote again. So what do I need to do to the betas for this model to represent a model of competition? Do I need to make them positive, negative, or I can set them to be anything and it's always a competitive model? Ready? 3, 2, 1. Yeah, okay. So I'd say that helped. Almost everyone turned to beta being positive now. And can someone say why? Positive beta decreases, yeah. So you see that, I mean, this minus is sitting outside here, right? It multiplies this whole thing. So positive beta means that the presence of the other species linearly decreases the per capita growth rate of the other species, right? This thing here is the effective per capita growth rate, right? And the simplest thing that you could have is a model in which there was a linear dependence between the per capita growth, of the effect of one species on the other's per capita growth rate. The same thing over here, right? You could imagine more complicated models where maybe it was quadratic or some fractional power. This is perhaps the simplest thing that you could write down, right? Okay, great. And so you can see that effectively, yeah, you could think about it as some sort of linear inhibition when beta is positive. What happens when beta is negative? Yeah, they don't have to grow, but their growth rate increases when in the presence of the other species, right? And so that would so if both the betas were negative, that would represent a mutualism, right? Both the species are helping each other to grow. And in that case, in fact, even if the R's were negative, if each of the species had a large enough negative beta, it might help the growth rate become positive, right? And so that's the sense in which I was hesitating to say that, you know, R negative does not always mean that species cannot grow. In fact, this is a kind of really interesting case where even in this simple model, in each other's presence, if the betas are strong negative enough, even with R negative, these species could grow, right? And so this would be a case where on their own, these species would die because their logistic growth rates would be negative. But in the presence of each other, their growth rates would be positive and they would grow. Right? And so this model really, by choosing the betas appropriately, can be a model of competition or a model of mutualism or a model of even a kind of interaction where one species competes with the other but also gets help from the other, right? So yeah, you can play with you can make one negative and one positive to get that kind of thing. Yeah. So yes.
Will we get the predator thing on if one of them is negative?
Yeah, you should try that out. You will see that you will get something not very different from one of the predator-prey models we wrote down in last class, and you can figure out which one that. Yeah, you have to choose the betas accordingly. Correct. Yeah. Okay. So in the rest of this, for the rest of this class, we're going to stick with competitive model where the betas are all positive, right? And there's also a very useful change of variables, which we're going to make, which is going to make our lives much easier and not make us carry around these Ks. So the change of variables that you make is that you normalize the abundance of each species to its respective carrying capacity. What does this mean? It means that x1 is now, the fraction of species 1 that's present in units of its carrying capacity, right? So x1 equals 1 would mean that species 1 is at its carrying capacity. x2 equals 1 would mean species 2 is at its carrying capacity. Typically, you might expect just with logistic growth for x's to take values between 0 and 1, which is the thing that we like. And so if you do this change of variables and rearrange things, you'll find that the same model in terms of the x's now looks much simpler. Just to write down. And this is the this has a few names. This is either the standard form of the Lotka-Volterra competition model, or it's the generalized form of it, and so on and so forth, right? And then you can relate alpha 1, 2 with beta 1, 2. And alpha 2, 1 with beta 2, 1. Right? So the carrying capacities, as you can see, will get absorbed essentially in these alphas. Right? And so now you'll see that the only things that we really need to care about are these alpha 1, 2 and alpha 2, 1. Okay, so this is the model that we'll work with for the rest of the class. Okay, and so we've been doing this for the last 2 classes, so by now you should have a handle on what are the things we should do once we encounter a model like this. What's the first thing we should ask ourselves? Yeah, the first thing before we even get to stability is we should ask ourselves if there are any fixed points, right? Okay, so let's ask that question. Let's ask how many fixed points there are, and we'll all vote. I'm going to make it simple for you. We're just going to work in a case where R1 and R2 are both 1. Okay, how many fixed points? Ready? 3, 2, 1. Okay, I would say many people are saying 4. Can someone give me the rationale for 4?
0, 0, 1, 0, 0, 1.
Yeah, so it's true. So x, so yeah, you could have 0, 0, that could satisfy, right? That of course is one that we know will work. We already worked out that when one of the species is not present, the other can be at its carrying capacity. So 0, 1 and 1, 0 will be fixed points. And then there's in principle a fourth one, right? And this fourth one will turn out to be a bit tricky. Because as we'll find out, it may not always do something, right? So maybe there's a fourth one as well, right? So what do I need to do to look at these fixed points? I need to set x1 dot to 0 and x2 dot to 0, right? And I need to satisfy both of them. So of course, one way to make that happen is for x1 and x2 to both be 0. What's the other way? The terms in the parentheses can be 0, right? And that gives us some conditions, right? First one gives me this condition. The second one gives me that condition. Right? And now I need to simultaneously solve both of these things to get what I need, right? So this you can sort of verify the 0, 1 and 1, 0 will do it. But this is some linear equations in 2 variables. In principle, you can also have a case where x1 and x2 are both non-zero. What are these equations of? Sure, it's the per capita growth rate. Okay, let me give you another hint. I have a two species system, so I can make a phase portrait, right? So I can make x1 over here and x2 over here. Right? Say or x1 equals 0 or x2 equals 0, right? And I want to geometrically look at these conditions on this phase portrait. What should I do? I should draw lines. Okay, why should I draw lines? These are equations of lines, right? And in principle, these are some special lines. They're called nullclines. I mean, nullclines don't always have to be lines, but these are curves where the following happens. This one, remember, is x1 dot equals 0. This one is x2 dot equals 0. Right? And you see that all 4 of these are lines. Right? Okay, so let's draw some of these. I need all of my colors now. Okay. So let me use the following convention. Let me use orange for x1 dot equals 0. It's another color. Let me use pink for x2 dot equals 0. Are these 2 colors distinguishable in the back? No? Maybe I'll erase these and use green instead. Okay. All right, so we can draw the two easy ones first. Each of these will give me an axis. X2 dot equals 0 should be x2 equals 0. That's this line, right? And this one should give me x1 equals 0. X1 dot equals 0, that's this line. And then each of these, I need to think about, right? They're lines. So let me draw one of them. Let me draw this one. This one is going to be some line like this, right? Why is that? Because when x1 equals 0, this is going to give me x2 equals 1 over alpha 2, right? So that point here is going to be 1 over alpha 1, 2. And when x2 equals 0, what will I get? x1 equals 1, right? Okay, now I want to make the second line. Okay, but here is where I'm stuck a little bit. Can I just make that line, or are there cases that I need to consider? I need to consider cases, right? So I'll just give you the following question and you think about it and vote on your own. How many cases do I need to consider? And you can take 1, 2, 3, 4, 5. Okay, is the question clear? I want to draw the second nullcline, just the fourth one that I haven't drawn yet. But I'm saying, I can't just draw it without knowing something more. There are 4, 5, 6, 7, some number of cases that I need to consider. Okay. All right, let's vote. Ready? 3, 2, 1. Do people need more time? Okay, maybe you can discuss with each other what you think the number of cases should be. Okay, let's take a vote again. All right, ready? 3, 2, 1. Okay, I'd still say there's a healthy mixture of 3s and 4s. So maybe we can have a little bit of a debate. Can someone with the 3s give an argument for which 3 cases? Alpha 1, 2 less than
Alpha 2, 1 and alpha 1, 2 greater than alpha 2, 1 and then both of them are equal.
Okay, I see. So alpha, okay, so what would each of those cases mean? What would happen if alpha 2, 1 were smaller than, less than, or equal to? I mean, we care about these lines, right?
The slope depends on alpha 1.
The slope depends on alpha 1, 2, right? So yeah, so here we know that this line passes through 1 and 1 over alpha 1, 2. We know that the other line also by symmetry should pass through 1 here and 1 over alpha 2, 1 here, right? That's just by symmetry, right? So now the 1 here could either be over this side or that side, right? Why? Because, well, I don't know what this alpha 2, 1 of this alpha 1, 2 is, right? So when would my line be below? When would it be above? Correct. So it depends on whether alpha 1, 2 is greater than 1 or less than 1, right? Yeah, so 1 is the appropriate comparison here, right? Here also, the point of this green line is going to be 1 over alpha 2, 1. And whether it's on this side of 1 or that side of 1 is going to depend on whether alpha 2, 1 is greater than 1 or less than 1, right? So there are 2 cases over here and 2 cases over here. So there are 4 cases that we need to consider, right? And the 4 really depend on alpha 1, 2 greater than 1, alpha 2, 1 less than 1, alpha 1, 2 greater than 1, alpha 2, 1 greater than 1, alpha 1, 2 less than 1, alpha 2, 1 greater than 1, and alpha 1, 2 less than 1, alpha 1, 2, 1 less than 1. Right? Both of them can be less than 0, both of them can be greater than 0, one can be less, one can be more. And based on that, I'll make 4 different kinds of lines, right? So, you know, maybe if I were to make those lines, one of those lines would look like that. One of those lines would look like that. One of those lines would look like that. And one of those lines would look like that. And let me label these lines so that we can do some matching. 1, 2, 3, and 4. And these are the 4 cases, right? Can we just quickly go around matching? Maybe I'll call these also things. I'll call these A, B, C, D. And let's just go around saying 1, 2, 3, 4 for each of these cases. So what does A correspond to? Which of these lines does A correspond to? So A is alpha 1, 2 greater than 1, alpha 2, 1 less than 1. Ready? 3, 2, 1. Okay, people need a little more time. Yeah, so I have the first case, which is A. I have 4 lines here. I want to ask which of those lines is the first case. Okay, ready? 3, 2, 1. Okay, very few people voting. So let's go through this together. So alpha 1, 2 greater than 1 means what? 1 over alpha 1, 2 is less than 1, right? So this point would be 1 always, right? So it can't be these two. So it can't be 1 and 3. It has to be 2 or 4, right? Now we have to look at the other case, which is alpha 2, 1 less than 1, right? And so if alpha 2, 1 is less than 1, 1 over alpha 2, 1 is greater than 1. And so that would mean this case, right? Do people agree? So that's 4. Yeah. Okay. What about the next case? What about B? When both of them are greater than 1? Let's take a vote. Ready? 3, 2, 1.
Actually,
Yeah.
it's a bit confusing. Like, can we draw the, one line and do like the line passing through 1, 1, so 1, 0 and 0, 1, then do all these.
Okay, I see. So you're saying that you want me to draw a line which is 1, 0 and 0, 1?
And then when we look at these cases, then it will be easy.
Okay, really? Maybe would it be easier if we went this way?
But here it is like we are taking one of the 4 lines.
Yeah.
And then we are,
Where is the 1?
Yeah, 1 and 1.
Where is the 1?
Does that really matter?
yeah, only then we can say if it is greater than 1 or less than 1.
Okay, I see. So look, we know that x2 dot, the point where it intersects here is always going to be 1, right? Do we agree? Look, by looking at this orange nullcline, you can see one of the points where it intersects is 1. The other one is 1 over alpha 1, 2, right? Similarly, the green line, over on this side is going to cut at 1. On this side is going to cut at 1 over alpha to 1. Right? So you know that whatever I've drawn, it's always going to be 1. Right? That's why it's not possible for me to draw the 1, 1 line. It depends on which cases we are considering. So I'm doing a funny thing, right? I fixed alpha 1, 2. So that means the 1 keeps changing as alpha 1, 2 keeps changing. Right? But okay, let's go through these together if this is being some trouble. So let's look at the line 1. For line 1, what would be the case? This point is 1, right? So 1 is less than 1 over alpha 1, 2. So alpha 1, 2 is, if it's less than 1, right? 1 is here. This is larger than 1. So 1 over alpha 1, 2 is less than 1. Right? Same thing here. Right? So this means both of the alphas are smaller than 1.
One of them is greater.
Okay, good. Yes. This one, alpha 1, 2 is smaller than 1. This one is greater than 1. Right? And so on and so forth. Right? And so this inspires us to make a phase diagram where we learn that really the things that we should care about are whether the alphas are greater than 1 or less than 1. And so what you can do is you can just plot log of both of these quantities on both axes. Right? And so of course when log of alpha 1, 2 is greater than 1, you're on this side. Log of alpha 2, 1 is greater than 1, you're on this side. Right? And so on and so forth. Right? So there are 4 possibilities, and these are the 4 cases. In this case, we'll see that there are these 4 qualitative there are these 4 things that you see. On this side, you see bistability. On this side, you see coexistence. On this side, you see that species 1 will win. And on this side, species 2 will win. And we'll go through one of these cases now to get a sense of it. And are there any other possibilities for the alphas?
Both of them are equal to 1.
Yeah, good. So if both of them are equal to 1, then you sit at the origin. And then,
whole nullcline is like fixed point.
yeah, good. And what would it mean in the model if both the alphas were equal to 1? Is it still a 2-species model?
It's equivalent to other individuals of the same species.
Yeah. So look, this alpha 1, 2 tells us how species 2 is inhibiting species 1. Right? And how much is species 1 inhibiting itself? By some amount 1. Right? There's secretly an alpha 1, 1 here. Right? And so when alpha 1, 2 equals 1, that means the species 2 inhibits species 1 the same way species 1 inhibits itself. Right? Similarly, when alpha 2, 1 is equal to 1, it's the same thing. Right? So from the perspective of species 1, it doesn't care, whether there's a little bit more of species 1 or species 2. It feels the same inhibition. From the perspective of species 2, the same thing. Right? And so in this kind of case, it doesn't really matter that there are 2 species. Right? And so in that sense, you get some degenerate case where both of them will survive, but it's like one thing surviving. Right? Okay, good. And remember, we're dealing with the case where both these alphas are positive. That's why, this log space will never allow us for the alphas to go negative, and we're not considering that case. Right? Okay, so let's try to see each of these cases. Okay, so let's work out one of these, orange and green. Let's try and work out the one with coexistence. why were we drawing all of these nullclines? We wanted to care about the fixed points, right? How would we see the fixed points from the nullclines? Let me make one of these cases now. Okay, all right. Okay, so did I draw it right? This point is 1. This point is 1 over alpha 2, 1. So what case am I in? I have alpha 1, 2 less than 1 and alpha 2, 1 less than 1, right? So that's the case that I'm in. And I know that by looking at that diagram, I should get coexistence, right? So how do I see by looking at this plot that coexistence is even possible? Is there only one fixed point? Right. So why do we draw these nullclines? How do they help us find the fixed points? Yeah, the intersection. Yeah. So remember, to find fixed points, we need x1 dot to be 0 and x2 dot to be 0, right? We know that each of these conditions is a line. So for both of the conditions to be simultaneously satisfied, we need lines to intersect. Now, do we need any lines to intersect? These two lines also intersect here. Can this be a fixed point? Ready? 3, 2, 1. No, right? Yeah, because it's the same species. This is x1 dot equals 0 and x1 dot equals 0. That's not a fixed point, right? Same with this guy here cannot be a fixed point. What about this here? It can be, right? That's our 0, 0 fixed point, right? So now you can easily read off by looking at wherever a green and an orange line intersect, right? There's one here, there's one here, and there's one here, right? And so there are 4 here. And so do all of these points mean coexistence? No, right? Only this one means coexistence. And of course, now you can see if I had a nullcline that went like here, then of course no coexistence was possible, right? Similarly, if I had another nullcline that went like that, I would have no fixed point or no coexistence possible. Okay, and so now I have this coexistence. We'll talk about its stability in a second. Let's quickly inspect the stability of these other things. What about the 0, 0 fixed point? Is it stable or unstable? We'll say thumbs up for stable, thumbs down for unstable. Ready? 3, 2, 1. Yeah, unstable. Can someone say why?
Use little of population 1.
How do I see that? Yeah, we know that in the absence of either species, you just have logistic growth, and we know for logistic growth, 0 is an unstable fixed point. So at least there are 2 directions in which you'll go away. And so that automatically means you're unstable, right? Okay. What about this one? What about 1, 0? Is that stable or unstable? Ready? 3, 2, 1. Okay, there seems to be some confusion or some mixture of people saying different things. So turn to your neighbor and try to talk about whether it's a stable or an unstable fixed point. Okay, let's turn back and vote again. So we're talking about this fixed point 1 comma 0. Is it stable or unstable? Ready? 3, 2, 1. Okay, I still see quite a healthy number of people saying stable, healthy number of people saying unstable. Someone with an argument for stable? I know there's many of you thinking that it should be stable, so someone should have an argument. Or did you toss a coin? Folks, it's very important to do the reading before you show up to class, otherwise you're not going to have a fun time. Yeah, so which of the cases are we in? Correct, it depends on the alpha.
1 minus alpha 1, first equation. So if alpha 1, 2 was less than 1, the slope is positive. So x1 dot is positive. Around.
Yeah, so should you be looking at x? Yeah, so okay, good. So first of all, we need to know what the alphas are, right? We know that. We know that both of them are less than 1, right? Second of all, we need to ask which direction we are assessing stability in, right? So okay, so let's talk about the easy direction first, which is the x1 direction, right? Is it stable or unstable in the x1 direction? Stable, right? And did we have to care about the alpha there? Why is that? Because x2 is 0, right? So if x2 is 0, then we only need to care about x1. So it's stable in this direction, regardless of the alphas. What about in this direction? Now I need to care about the alphas, right? So what am I doing when I'm going a little bit up? I had a condition in which x1, species 1, was at its carrying capacity, right? x1 was equal to 1. Now I'm adding a little bit of species 2, and I'm asking if that grows or not, right? Remember, that was the definition of stability that we were even talking about in the beginning of this class. So let's put a little bit of species 2, right? Species 2 has a growth rate that depends on this bit, right? Add a little bit of that to what steady state? To a state where x1 is 1, right? So now the growth, I mean, x2 of course is going to be positive, some small positive number. And what should I care about for this term to know whether it'll grow or not? Yeah, I need to know whether it's positive or negative, right? And x2 is very small compared to 1, so I can ignore that. So I just end up with 1 minus alpha 2 1, right? If 1 minus alpha 2 1 is positive, this thing will grow, and you'll get instability, right? And that's the case. We know that alpha 2 1 is less than 1 here, right? So in this case, you'll go up, right? And you'll get this to be unstable. Okay, can you quickly try and check the same logic for this fixed point? We'll vote in 10 seconds about whether this one is stable or unstable. Remember, both the alphas are less than 1. Okay, so is this fixed point stable or unstable? Ready? 3, 2, 1. Unstable, right? Same logic. It's stable in this direction, but as soon as you add a little bit of species 1, you have to worry about the growth rate of species 1, and that's going to depend on alpha 1, 2 being less than 1, and it is less than 1, so it'll grow. So thus far, all of our attempts at finding a stable fixed point have been unsuccessful. We found 3 out of 4. We found them all to be unstable. This is our last hope. If it's not, then we go to our Kolmogorov conditions and Poincaré-Bendixson, etc., etc. Let's see whether we need to do that. How do we check here? Right, so one thing you could do is what we've been doing in the previous classes, which is to evaluate this fixed point and find the Jacobian around this fixed point, and then do what? You look at the eigenvalues and see if they're positive or negative. And in this case, do you know what you'll find? They'll turn out to be negative. And so this one, this fixed point is going to be stable.
If the Jacobian doesn't have eigenvalues, then
If the Jacobian doesn't have eigenvalues, it just doesn't have. When would that be?
something like 1, 0,
Do you
1, 1, 0, 1, the matrix doesn't have. Just one eigen.
Yeah, I mean, you'd always be able to find the characteristic equation. You'd always be able to look at some solutions of it. Sometimes they might not be real. Sometimes they might not be something or the other. But just by looking at the real part,
You will have a single eigenvalue.
Yeah, that's fine.
So suppose you have the matrix 1, 1, 0, 1.
Yeah,
The characteristic equation is x minus 1 whole square is 0.
yeah.
So it has just one eigenvalue 1.
I mean, there are two degenerate eigenvalues,
There's no
right?
degenerate eigen. Just the vector 1, 1, 1.
What do you mean just the vector 1, 1?
So the vector 1, 0 is an eigenvector,
Yeah.
and there's no other eigenvector.
Yeah, but what about the eigenvalues?
It's just one.
Yeah, so that's fine. And so this one, you perturb it along that direction and it'll go on, right?
But in the other directions?
Yeah, that's fine. But remember, for instability, you just need one unstable direction.
But if you want to prove it's stable,
Then you need to show that along all directions you can perturb it and nothing will happen. Yeah. Okay. So in this case, you will end up with a stable fixed point, and in this case, these species were coexisting. So you'll get stable coexistence. And that, in fact, is that outcome on the bottom left that we were looking at. Okay, say that I didn't want to invoke this Jacobian for a second. Is there any other trick I could have used just by looking to see whether it should be stable or unstable?
We look at the nullclines. If we're above one species nullcline, we know that the rate of change, the derivative basically should be either it should be negative. Like for the green nullcline, if that is x2, but so if we go up, we know it should point down. So
Why are you saying that? What about on the nullcline itself? So I drew these helpful arrows for you. Is there something about the directions that the trajectories will take on the nullclines themselves? So x1 dot equals 0 means that you can't have any flow pointing in the x1 direction, right? So all along this line, your flow will either be up or down, right? And so here we just calculated that it should be up. It'll be up, up, up. Oops, sorry. At some point, it'll become 0, of course, at this point. And then you'll change and it'll be down, down, down. The same thing for the other nullcline, right? This is x2 dot equals 0, so there should be no flow along the x1 direction, the x2 direction. And so this one will go right, 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 and it'll change in flip direction, will go left, left, left. Right? And now you can ask, okay, what happens if you go a little bit above? If you have a little bit below, then these things will start to curve, right? And so now in principle, you can see, you might be able to see that here you will have trajectories that will kind of do something like this. Yikes. Right? So even by looking at some of the arrows on these nullclines, you can try and guess whether the flow should go into the coexistence fixed point or not. And so you'll see that a funny thing that will happen when you take this other case. When alpha 1, 2 is greater than 1 and alpha 2, 1 is greater than 1, you will also find an intersection. In fact, I'll just draw it here for us to see quickly. Right, so you'll get one line that looks like this. And these lines will still cross, but those ones are crossing from above, these ones will cross from below. You will get this kind of situation. Right? This one is 1, this one is 1, this one is 1 over alpha 2, 1, this one is 1 over alpha 1, 2, right? And now you still get one of these fixed points, right? You get this guy here, this guy will be unstable. You get this, you also get these ones. And so you also get this and that. Is this clear how I got these 4 fixed points? I am again asking whether 2 lines of different colors are intersecting. So this is not a fixed point, that's not a fixed point, right? So this is also a coexistence fixed point, right? Should this also be stable? So now you can do the Jacobian analysis for this case, and you'll find that it'll be unstable. And you can also kind of see that this time the arrows will flip. So this will now have arrows pointing up. This will have arrows pointing down. This will have arrows pointing to the left. This will have arrows pointing to the right. Which way do the arrows point here? At this point, they point nowhere, right? It's a fixed point. Okay, and so now you can also see that flow kind of goes away from this fixed point. And so it's actually unstable. Yeah. Perfect. Yes. So now the question is, when the flow goes away from this point, where do you go instead, right? And so, I mean, I erased it over here, but remember we had this invasibility condition of 1 minus alpha 2, 1 and 1 minus alpha 1, 2 being greater or less than 0. You see when both the alphas become greater than 1, both of those will give you stable fixed points, right? So now in this condition, both of these points will become stable. And so what condition does this correspond to? This gives us a bias stability, right? So that's this condition. And indeed we can see that both of the alphas are greater than 1. So we see something like this. So these flows will kind of take you towards this fixed point. So what about this coexistence? If you actually start off the system at this point, it will stay there forever. But if you perturb it even a little bit, it will go either this side or that side, right? And in fact, you can draw a separatrix here. Which is essentially a line joining both of these unstable fixed points. And you'll see that basically all the trajectories on this side of the separatrix will take you to that stable state. All the trajectories on this side will take you to that stable state.
How did we know that the separatrix was stable?
Yeah, that's a good question. You can try and work that out by looking at the eigenvectors of the Jacobian in this case. And you'll find that one of the eigenvectors will be stable. So now you can use your eigenvector argument to find exactly that direction. And then you still won't know it's a straight line all the way through, but now you can show, you can find you can find the equation of this line, plug it into your equations and see that actually it's a line that separates both of these things. So actually along this separatrix, your flows will run into this fixed point. Yeah, good question. So remember, the nullclines themselves mean either x1 dot is 0 or x2 dot is 0, right? So when x1 dot is 0, that means that whichever way your flow line is pointing, it should have no x1 component. No x1 component means either vertical, up or down. Same thing for x2 dot equals 0. So just by definition, the nullclines will either be vertical or completely horizontal.
And the direction is decided by the nearest
By the direction, you'll kind of have to ask something clever.
After half the line,
Yeah, so you know here it will become 0. So if it was positive on this side, it will be negative on that side. It's a
unstable fixed point near that is near to that the direction should be 0.
around any fixed point, the direction should change, right? Because you knew your nullcline was a smooth curve. If it crosses 0, on one side it will be positive, on the other side it will be negative. Right? And we know in this case the nullclines are all smooth curves. Okay? And so this way you can also work it out for the other two cases that I haven't worked out here. In this case, you'll see that these nullclines actually won't intersect at all. And in that case, you'll be able to show that there are actually only 3 fixed points, and one of them is stable, 2 of them are unstable. And which one is stable will depend on these different conditions, right? And so let's spend the last 5 minutes just talking about what we did. We started the class by talking about these 3 qualitative outcomes that we saw, right? What were the 3 qualitative outcomes? We saw exclusion, where one of the species wins. We saw coexistence, where both species can coexist stably. And there was bistability, where one or the other wins depending on the initial conditions. And there was this zoo of other things that we talked about could possibly happen, but we don't see, right? And then we made a model, we asked about competition, and then we studied various things about it, and we found that, okay, there should be these things that should be happening in this model. So did we win? Did we lose? How do we know if we won or not? So, okay, so how many qualitative outcomes did we see, or does this model have? Yeah, you might say there are 4, but I mean, species 1 winning or species 2 winning is just a relabeling thing, right? So in principle, we see 3 outcomes, right? And these are the 3 qualitative outcomes we talked about we see most often in experiments. So I don't know how you would classify it. I would classify it by saying we kind of won, right? And this kind of also maybe tells us a little bit about how in this model, it's probably not possible to see these kinds of outcomes, these other kinds of outcomes that we talked about, right? For instance, you can try and work it out at home, what it might take to give you a condition where you have a coexistence fixed point and another stable fixed point where one or the other species wins, right? And you'll see that you can't construct. Okay. The last thing I'll say is you can construct such a scenario. Can someone, okay, yeah, good. Can someone guess how?
Some nonlinear curve.
Correct, yeah, exactly. So in this case, we dealt with nullclines that were all straight lines, right? And why were they straight lines? Yeah, because we wrote down this simple Lotka-Volterra model where the inhibition from species on each other was linear in their abundance, right? You could write down another model which was nonlinear, which would give you nullclines which are curves, right? And then you can go in your notebook and try and work out, this kind of phase portrait for curves, and see that there actually you can construct scenarios. And in fact, you should even be able to construct a tri-stability, which is what Jitendra was talking about. So see if you can work that out. So just a small hint for you is, in all of this, we were thinking about the single species being logistic, growing logistically in the absence. In this case, you might need the single species growing with an alley effect. So if you introduce a nonlinearity in both the species on their own by an alley effect and species affecting each other in some non-linear way, you'll be able to construct scenarios where you can see tri-stabilities and so on and so forth. Right, and then you know exactly what kind of species to do these experiments with to see those kinds of outcomes.
How do you estimate these parameters in an experiment?
Yeah, that's a wonderful question. We'll talk about that in the next class. What I'll quickly say is that, look, now we did this experiment and we got the dynamics of what the species abundances with time look like, right? You can try and fit your model, fit the dynamics to your model and try and guess what the alphas can be quantitatively. Qualitatively, just by knowing the outcome, you should be able to tell which part of the phase space you sit in, right? You should at least be able to say whether the alphas are greater than 1 or less than 1. And if you knew the carrying capacities, and you'd also be able to figure out their bare betas. Okay, so in the next class, which is the last class of this module, we will generalize this model to many species competing, and there we will ask the question of stability, which will involve a Jacobian which doesn't have 2 eigenvalues, but has many eigenvalues, which will mean that we want to talk about distributions of these eigenvalues, and there we'll talk about stability or instability in terms of the distributions of these eigenvalues. And we'll see something quite interesting, which hopefully will be thought-provoking, and then we'll end. All right. Yeah, and that's the end of today's class. I'm going to stick around for the next 25 minutes or so. If any of you have any doubts, you can come to me. The rest of you should feel free to prance around.
For the other than single species, there are bicyclic conditions coexistence.
Yeah.
So there, if you pick by the species, this should happen only if they fight or something like that, right? It just won't happen because of the nutrients. Because I mean, if food is the only thing, only they are fighting for, or like they're searching for food. Whoever is able to find food most effectively could win. That could result.
So you're talking about, so you think that biostability might not be possible if nutrient competition was the only thing that's going on, right? That's a great question. So I mean, you're right that maybe one might intuitively think that, but it turns out not to be true. And if you're interested in that, you can come to me, and then we can talk about offline. You can write down like a resource explicit model, the one that we didn't write down, and there you'll see that there are some conditions in which you can get biostability, but I can't provide you a very intuitive explanation as of now. For that, you really need to understand what that model looks like. Yeah, but the key is really in the fact that when you're eating nutrients, there are 2 things that you're doing. One is that you're depleting the nutrients that you're eating. The other is that you're growing as a result of that. And both these things can be slightly different from each other. And it turns out when both these, there'll be some coefficients that determine this. When they're different enough, you can get instability. You can get biostability. And that's because I'm eating something that I'm not depleting as much, or I'm depleting something a lot that I'm not growing as much on. But by eating a lot of it, I'm not giving the other species the opportunity to eat it and grow. That's kind of how you can get a biostability. So whoever starts more will deplete all the nutrients quite early, not grow too much as a result, but it'll survive, but not allow the other species to make it the other way.
If we don't consider resource competition at
Yeah,
all,
yeah.
can we still say that this inhibition coefficient data that we've considered has implicitly captures the resource inhibition aspect?
Yeah.
Because so if when we rewritten the equations, so it was beta times the number of the other species.
Correct.
Number of the other species would increase if it's taking in more nutrients.
Correct. Yeah, so
Implied in a way?
well, I would say the model implies is what it's trying to say is that it's agnostic to the mechanism of the competition. There could be many things as long as they are density dependent in a linear way. You don't need to care about them. But you're absolutely right that if you try to write slightly more mechanistic models of the resource explicit kind, for instance, the kinds that are at the end of the reading, then you can explicitly try to show that there are regimes in which you can write down the beta in terms of the resources. And there also you can show that you can go from that model to an effective Lotka-Volterra model, where you can write down the beta as exactly as some number times resource concentrations. Yeah.
So if there are n species, for each pair of species there is this interaction term.
Correct.
Beta.
Correct.
Would that be same as the interaction term if those two pairs are in isolation?
That's a very deep and nice question. So in these models, in the Lotka-Volterra models, that is indeed what you will assume.
So if it's like physics objects like charge or masses, I can say it's same.
Yeah.
But since it's ecology,
Yeah.
there can be other kind of interaction we don't know.
Absolutely. So you're absolutely right that when you have the same two species and you put them in a totally different environmental context, they might compete with each other very differently, right? And it might be that all the other species' effects on the environment also show up, right?
So for all the practical purpose, do you take each pair out from the combination and find the beta? And for all the calculation, do you use those beta values?
Yeah, so if you're trying to study a Lotka-Volterra model and fit it to data, that's exactly what you would try to do. But like I was saying before, you could also just circumvent the problem of by trying to explicitly introduce resources into a model. So there, the interactions become emergent quantities that depend on how the resource competition is going on, and that will depend on what other species are there. So then your beta, your set of betas themselves become dependent on the state of the system. They don't become fixed numbers. Yeah, so I mean, for that we really need to study the what are called consumer resource models. But the way you would calculate it, just without going into it, is that you would work out a model where there are resources and species. You have dynamics for both sets of these things, and you will try to ask, are there limits in which that model looks like an effective Lotka-Volterra model? And there might be limits where it looks like that. And then you'll ask, okay, the beta that I got now, what does it look like in terms of all the other underlying parameters that I have?
Resources.
Yeah.
There are like 10, 20 species.
Yes.
The pairwise interaction is different now.
Yeah.
How will you calculate that?
Yeah, but there the pairwise interactions will all be mediated through resources, right? It's either because we are eating resources or I'm producing some resources. So just by knowing what the resource environment is, I can know how you will affect me.
It can be a direct interaction.
There can also be a direct interaction, yes, which you will have to put in some Lotka-Volterra way rather than in some resource explicit way. Yes.
You spoke about there being something more subtle to carrying capacity, not entirely depending on the resource.
Yeah.
Could you elaborate on that?
Yeah, okay. It's okay. From what it's sounding like, you all want to actually study consumer resource models. Okay, just for lack, so I do teach this whole course at ICTS on ecology and evolution, where I do both of these things. But because of lack of time, I only chose one. The elaboration of the carrying capacities is again, if you start off with the resource explicit model, and you try to massage all the terms to make it look like a Lotka-Volterra model, you'll find an effective carrying capacity in terms of the underlying parameters as well. And what you will find there is that itself is a dynamic quantity that depends on the resource environment and on various other things. And so in that way, the carrying capacity itself depends on resources, but it's not like a fixed quantity. There's some question at the back, and then I'll come to you. Yeah.
As wondering about pathologies, I mean, you described two species here.
Yes.
Let's say I don't know how to differentiate between species. Let's say you just gave me a sample.
Yeah.
And maybe there's only one species.
Good.
And how would I model? Let's say there are two populations.
Yes.
Using the same bio.
Yes.
I'm confused by which. I mean, in principle, I'll have to just write one equation.
Yes.
But since there are two populations in my head,
Yeah.
I'll write two equations and definitely answer won't work out. Because it's probably not describing the correct growth rate and other. So, I mean, this is slightly confusing.
Yeah, great question. So actually what you're describing is not hypothetical at all. This actually happens in microbial populations where there's this growing understanding that what we think of as one species actually has some heterogeneity within it, where there could be some subpopulations. Now the question really that is, I would say, kind of state of the art is how different are those two subpopulations in how they grow, how they compete with other populations, right? And I would say that the answer to that is not clear at all. So you can imagine now one regime where those two subpopulations actually behave very similarly. And if they behave exactly the same, then the one single equation that you write down for the one species will be a very good approximation. The more and more these subpopulations become different, the less and less accurate your model will be. The problem with fitting this model, of course, right now is with just this, that you'll see some outcome for the combined species, and you can for that outcome you can fit an alpha, right? So it'll always seem like you can fit an alpha for a two species outcome. And the question that you really need to ask is how generalizable is that? Right? When you have now 3 species or 4 species or 20 species or 100 species, how well do those alphas work? And maybe in those conditions, those two subpopulations, when differences between them will really start to become stark. So I think this question of how different are these subpopulations in terms of how they compete with everything else in the community is an unsolved one. So I didn't fully answer your question, but it's because we don't know the answer to it yet. But at least mathematically, we can kind of see when it should work and when it should not work. There's another question.
I have a question. Like, could you share some mechanistic models?
Yeah. Sure.
The question is, like, this is just for 2 species, but in nature we have many species, and
correct.
We just think of, in terms of n species, and just 2 states extinct or stays or goes away. You see exponential growth in the number of possible attractors.
Absolutely, yeah. There's exponentially many states. That's exactly,
so like, in gene networks, at least, I know of statistical tools to study those. Do these extend to ecology, or I don't know. Yeah, so I know exactly which statistical tools that you're talking about, but in ecology also we have lots of tools from StatMech and disordered systems that we use to study these kinds of models in the limit where the number of species goes to infinity. Could you share that with me? Sure. Yeah, so what I'll share will have all of these things. Yeah.
Yeah. We talked about these like 2 species interactions on a very largest time scale as well, right? Like you have grasses as well in the initial.
Good. Yes. Yeah.
But we have a very large time scale. Wouldn't those species evolve themselves to like accommodate
absolutely
climate change? How would you like account for that?
It's a big open question. We don't know how to do it. So this whole endeavor is called eco-evolutionary theory. And again, when I teach my course in the first class, what I say is that we're going to do ecology and evolution. I do 2 modules, and I tell students explicitly that, look, I'll tell you now, but you'll forget it again. When we do ecology, we'll pretend as if evolution doesn't exist. And when we'll do evolution, we'll pretend as if ecology doesn't exist. And it is true. This is how both of these fields are at the moment. And we know that in reality, there's both processes that are happening. And so true eco-evolutionary theory is kind of absent right now. We don't have one. And that's one of the frontier areas of research in ecology and evolution. That being said, what I'll say is that what most people will give as a defense right now, especially in terms of trees and animals and so on and so forth, is that there's a separation of timescales argument that you can make, which is that ecological timescales seem to be fast compared to evolutionary timescales. In microbes, this is absolutely not true at all, as you said. So there, this excuse won't work. Actually, there's a lot of growing evidence that even for trees and animals, this excuse doesn't work. We know that, I mean, humans have evolved over the last few decades in many, many traits that we have. Yes. Oh, sorry. Yes.
Professor, could I ask a question?
Yes, of course.
So I had a couple of questions. First, so in this model, we've sort of assumed that when you have bistability, one species is sort of excluding the other, right?
We haven't
But
assumed that, right? We got that out. Remember, we
I
can.
mean, yeah, we showed that. But say if we had a situation in which the species sort of partitioned the resources between them and they sort of shared it, how would we sort of fit that into this kind of a model?
Right. So you're talking about some mutualistic scenario, right?
No, it's still there is competition, but like say there's like a range of sizes of the nutrition which they are, and like one species chooses the bigger size and one species chooses the smaller size, so that they like reduce the competition between them.
Okay, good. So what you're talking about has a name. It's called niche segregation or niche separation. And in that case, there's no competition at all, right? Both these, the big species is eating one set of things, small species is eating the other set of things. They don't affect each other's growth rate at all. And so, yeah,
Right.
they exist for all you care. Yeah.
But we couldn't show how that could also be like a fixed point
Well,
which the competition is driving towards.
Yeah, because we didn't write a resource explicit model, right? Again, if we had written down a resource explicit model, we would have explicitly been able to say what you're saying. Species 1 eats one thing, not the other thing. Species 2 eats the other thing, not the first thing. And then we would have been able to solve that model, look at its fixed points, and seen that you get a coexistence, a stable coexistence fixed point where they are not competing with each other.
Okay. And another thing was the separatrix which we drew in the bistability model.
Yes.
It's a straight line close to the fixed point, right? But away from the fixed point.
It passes through two fixed points, the origin and the coexistence, the unstable coexistence fixed point.
Right. But if you wanted to say find the shape of the separatrix far away from those fixed points, how would we go about that?
Yeah, good. Okay. So what I would do is, okay, so, if you would do what we do most of the times, which is that you would start off with your guess of the separatrix, which would be your stable eigenvector direction, and you'll plug that into your equation and see whether that works or not. And you'll find that it works. If it doesn't work, if it's not a line, if plugging it in doesn't work, yeah, then you're a bit lost. You'll have to guess something good and hope that it works. There's no general way to find it, as far as I know.
Okay. Thank you.
Yes.
In one of the cases where even one person could survive.
Yeah.
1% of the population.
Yeah. You mean when you add 1%, that's able to overtake and yes.
Or go to a fixed point.
Yes.
Survive.
Yeah.
I was talking about survival probability in the sense that if you just start stochastically, it's possible that one person will die off. So
yeah.
What kind of noise does the system? I mean, if you want to model it absolutely, what kind of noise is a good way? For example, it will be my Gaussian, right?
Yeah. So the kind of noise that is really really we care about is called demographic noise, which is a birth-death noise. And I'll tell you what it looks like, but I also first gave you the sense of where it comes from. So the idea is that when you have populations, in all of these models, we are modeling a birth-death process, right? This per capita growth rate was a combination of a birth and a death rate. And so when your populations are small, there's some stochastic births and deaths that are occurring. if there's a rate or a probability per unit time of each of those occurring, what you've written down is essentially some mean rate, the mean birth minus death rate, right? That will now have fluctuations which you'll have to care about. So if this is, for instance, a Poisson process, you'll see fluctuations which have variance equal to the mean, and so which will give you a standard deviation equal to the square root of the mean, and so you get basically square root n noise. So if I were to even write down, n1 ni dot, this is something what I would write down, blah blah blah. That's your deterministic part, and then I would add some root n, let me not call it beta, gamma times eta of t, where this is Gaussian white noise. Right, and so now you can see that basically as this population size gets smaller, the magnitude of the noise gets larger, and that's when you can stochastically, if this is sufficiently negative, your growth rate can trump that bit over there, and you can stochastically go extinct. So that's the kind of noise that people typically think about in these situations.
Building on this,
yeah.
Slightly continue.
Yeah.
We generally assume bacteria do Poisson, either divide over with the exponential rate.
Yes.
Right. Are there realistic models where, are there models where it's not really exponential? Maybe I would expect something like a Gaussian or something. बहुत जल्दी reproduce नहीं कर सकता, बहुत late reproduce नहीं कर सकता. There's a typical time scale where you would like to divide. In that case, can we actually still write this kind of models?
Good. Okay, that's a good question. I would say the kinds of models that you are talking about are ones where population sizes are small, right? So you really need to care about whether an individual is doing something or not, right? And those are very different kinds of models, and people do worry about them and write them. I would say the kinds of extensions of models that we write down over here are typically, we are interested in population sizes which are typically large, right? So in that case, the kinds of noise that we worry about is of this kind. There are other models that people write down where you can really try to incorporate this kind of noise. But typically then what you would do is that you're really writing down a stochastic process, right? And so then
If you write a mean field version of those things, is it okay to assume a model like that?
A mean field version of that? That's a good question. So there are models such as, there's one called Richard's growth, there's one called Compert's growth, where you can work out mean field things. There the noise actually doesn't look like this. It turns out the noise is not root n dependent. It's something called seascape or environmental noise, which is actually n dependent. And the reason for that could be, for instance, effectively what you can imagine is happening is that it's called environmental noise because you imagine the environment is changing. And because of that, if this becomes n dependent suddenly, now it's as if your R itself is stochastic, right? R plus some noise, right? So your growth rate, your per capita growth rate itself is a stochastic thing now. So there it turns out in those kinds of models, for reasons I don't want to get into in this class, you get a different kind of noise if you try to work up and derive the mean field models. Any other questions? Okay, great. Thank you. We'll meet for our last class of this module on Friday.